I am Impana and my team members are Sandhu Suresh, Gagana and Lekana HP. The title of our project is Brain Tumor Classification Using CNN, that is Convolutional Neural Network. We are working under the guidance of Professor Priyanka ma'am. Now, let's begin introducing our project. A project mainly concerns about brain. Brain is one of the largest and the most complex organs in the human body, which is made up of more than 100 million nerves. The main function of the brain is it controls and coordinates the actions and the reactions. Moving on to the main topic of our project is the brain tumor. A brain tumor is a collection of the abnormal cells that grows in the brain. The abnormal cells may become 2, 4, becomes 8 until there is a lump of abnormal cells. Brain tumors are classified into two types, that is benign and the low grade. Secondary brain tumor, which is known as malignant and also classified as high-grade brain tumor. Benign brain tumor exert pressure on brain causing headache. So headache is the main symptoms that a person show if he has a brain tumor or not. Moving on to the literature survey. Literature survey is going to be explained by Sandhu Suresh.
Good morning, everyone. The title of the paper is Brain Tumor Classification Using Convolutional Neural Network. Authors J. Sita and S. Selvakumar Raj. Published in the journal Biomedical and Pharmacology Journal. Published in the year September 2018. The main objective of this research work is to design a The title of the project is Brain Tumor Classification Using Convolutional Neural Network. Authors are J. Sita and S. Selvakumar Raj. Published in the journal Biomedical and Pharmacology Journal. Published in the year 2018. The main objective of this research work is to design efficient automatic brain tumor classification with high accuracy and with high performance and with low complexity. In this work, efficient automatic brain tumor detection is performed by using CNN. Simulation is performed by using Python language. The accuracy is calculated and compared with all other methods. In the proposed CNN, basic classification does not require feature extraction steps separately. Classification results are given as tumor or non-tumor, which contains sequence of feed-forward layers. ImageNet database is used for classification, so the training is performed for only final layer. Gradient descent based loss function is applied to achieve high accuracy, and the achieved training accuracy is 97.5%. Similarly, the validation accuracy is high and validation loss is very low. The drawbacks of this work is the automatic brain tumor classification is very challenging task in large spatial and structural variability of surrounding region of brain tumor. The next paper is Multi-classification of brain tumor image using deep neural network. Authors are Assam H. Sultan, Walid Atabani. published in the journal IEEE, published in the year 2019. Brain tumor classification is a crucial task to evaluate the tumors and make a treatment decision according to their classes. Deep learning is a subfield of machine learning and recently showed a remarkable performance, especially in classification and segmentation problems. In this paper, a deep learning model based on convolutional neural network is proposed to classify the different brain tumor types using two publicly available datasets. Presented a CAD system for the classification of brain tumor MRI images into three types in one study and further classification of datasets into different grades using CNN. The proposed network is constructed from 16 layers, starting from input layers, which holds the preprocessed images passing through the CNN layer and their activation function. The proposed CNN method is a segmentation-free approach. As we load the tumor image, get the corresponding slash directly, and the achieved accuracy is 96.13% using CAD system and 98.7% using CNN, and achieved 100% accuracy after 5000 iterations. The next paper will explain by Simpan.
Good morning, respected teachers. I'm Lekana HP. The title of the project is Multi-Class Brain Tumor Classification using GA and SVM. The main aim of this project is to develop a CAD system for assisting the radiologist in multi-class classification of brain tumor. A new hybrid machine learning system based on genetic algorithm and support vector machine for brain tumor classification is proposed. The texture and intensity of the brain tumor are taken as input for the model. They use 5 subclasses of brain tumor for the experiment. They use 428 data sets of 55 different patients. They collected the data set. Methods are used in this project. First, they collect the data set and they take it as input. Next, they extract some features from the dataset, just like gray level, co-occurrence matrix, Gaussian vector, and intensity-based features. Next, they select the features using GA, genetic algorithm. Here, the selected features which produce the best possible reset. The next, classification of brain tumor. The optimal set of features are selected by the genetic algorithm is used as input to SVM. Here, MATLAB software package 8.0 is used to perform the experimentation. Is classifier. The SVM used for classifier. SVM performs classified by pointing the data points into higher dimensional hyperplane through kernel tricks. SVM matrix means support vector machine as supervised learning model with associated with learning algorithm. It is used for analyze the data for classification and regression analysis. My observation and conclusion of this paper is here they develop a new hybrid GA SVM is proposed. The experiment consisting of mainly 5 types of subclasses of brain tumor that has primary and secondary that have subclasses, and they are different in aspects like their appearance, location, and size and shape of the tumors are different. Overall accuracy they get is 91.7%. Moving on to the next paper. The project title is Brain Tumor Classification Using Convolutional Neural Network. For most, they are the same method like the doctor has to inspecting the MRI images for the patient. For large amount of data and different specific types of brain tumors, this method is time consuming. So they are decided to for in this project they are planning to attempt a CNN model that can easily recognize and classify the three most common type of brain tumor. They implemented the simplest possible architecture of CNN, that is max polling, flattering, and layer followed by full connection from one hidden layer. For this project, they use 364 P1 weighted MRI image. Although they use only 700 images from types of three different tubes, and they use 500 datasets for training and 200 datasets for testing. The CNN architecture. They have followed three main steps. First one is convolutional neural network. CNN layer is a network where an image will be convoluted with filters to produce feature maps. The maps will be forwarded to the next convolution. Here, the layer receive and extract another higher level feature from input image. Max polling usually used as the downsampling operation, as it reduces the dimension while preserving the dominant features. Next is move to the convolutional layer. Flattering layer exists to vectorize the feature maps in the neural network. In the training phase, CNN use a learning or optimizer algorithm to update the filter at the convolutional layer and weights the neural network. The learning algorithm and takes a classification error or lose as an input and back propagates to the error into the network and update the filters. Next, the architectures. In CNN well-defined architecture, they use 5 different architecture like AlexNet, VGG16, RESNet, and RELU. Next, hyperparameter optimizer. Hyperparameter optimizer is a parameter whose value is used to control the learning process. Moving on to next paper, the title is Brain Tumor Classification and MRI Image Using Convolutional Neural Network. It was published in the year 2020. The main objective planned by the author in this paper is to classify the brain tumor various stages using CNN algorithm based on brain MRI images. And the parameters which is used for experimental setup is a convolutional neural network, MATLAB, and the OpenCV. The main contribution of the paper is that proving that the CNN image classification method is better when compared to other machine learning classification methods. In this paper, the datasets containing tumor and non-tumor MRI images are collected from different sources where MRI directly affects the treatment of patients, which highlights the image with the several colors. Next step is to improving the quality and removal of noise, resizing of image, and finally by using feature extraction method, the classifying the items according to its type and selecting the best classifier result. The conclusion of this paper is the CNN achieved the maximum accuracy of 98% when compared to other algorithms. And my observation in this paper is the use of CNN for classification of brain tumor within the given time by reducing the process time can be a successive model to achieve the goal. Moving to next paper, the title is Classification of Brain Tumors from MRI Images Using a Convolutional Neural Network. In this paper, the objective planned by author is to present a new CNN architecture for brain tumor classification and to test the network performance using several approaches. A comparison with the compatible state-of-art method is also done here. The parameters which is used are the dataset, image pre-processing, network architecture, and the main use of GPU, that is the graphical processing unit. The original contribution in this paper is firstly to examine the classification of different types of tumors from an imbalanced data set with a CNN and to show that the performance of the small architecture could also compare a variable with the performance of the more complex one. In this paper, the image from the database is taken that undergoes MRI image. Then the image pre-processing also takes place, in which transformation of image rotation can also be done. Next, creation of network architecture using a CNN that uses for tumor classification. Finally, the training network where randomly divided data into equal portions so that each tumor category was equally presented in each portion. The conclusion of this paper is a new CNN architecture for brain tumor classification was presented, a comparison with the compatible methods which also shows that the network compared better results. Keeping the architecture simple, classification in the real time could be possible and can examine the performance of designed neural network on other medical images also. And my observation in this paper is, as the proposed system in this paper is simple in creating a capable architecture for identifying different types of brain tumor, the network layers also can be detected. Moving on to next paper. Next paper is going to be explained by Gagana A.
Myself, Gagana. Paper title: Multi-classification of brain tumor from MRI image using convolution neural network with fully optimized framework. It was published in the year 2021. In this paper, brain tumors are classified into two types: malignant brain tumor and benign brain tumor. Benign brain tumors are usually tumors that develop inside the skull but outside the brain tissue. Malignant tumors are abnormal users that reproduce in an uncontrolled and irregular manner. In this paper, the disadvantages of this diagnostic method are that they are invalid, time-consuming, and open to sampling error. In this paper, we have to study about main objective is to designate three fully optimistic CNN convolution neural network model for multi-classification of the brain tumor. First attempt of multi-classification of brain tumor from given MRI images using CNN. The rest of this paper is organized as presenting related study and a detailed review of this study. In this paper, we have to classify 4 different data sets. The first data set is represented reference image dataset to evaluate therapy responses. Total number of images in this dataset is 70,220. Next step is represented repository of molecular brain neoplasia data. Third dataset is also cancer glioma atlas low-grade glioma. In this paper, CNN includes 5 main layers: input layer, convolution layer, pooling layer, fully connected layer, and classification layer. In this paper, we have conclusion. Structure and complexity of the CNN model vary according to the type of problem, input, and expected output. And moving on to the next paper. This paper titled Classification Using Deep Learning Neural Network for Brain Tumor. It was established in the year 2017. The main goal of this paper is brain tumor is one of the most complex organs in the human body that works with billions of cells. Brain tumor classified into two types, benign and malignant tumor. Benign known as low grade and malignant are known as high grade. The contribution of this paper by applying the deep learning concept to perform an automatic brain tumor classification using brain MRI images and measuring its performance. In this paper, proposed methodology based on the DNN, Deep Neural Network Learning Architecture for classifying were data acquisition, image segmentation, feature extraction, and reduction classification. The main conclusions of this paper, we proposed an efficient methodology which combined the discrete wavelength DWT with the deep neural network to classify the brain MRI into normal and three types of malignant brain tumor, glioblastoma, sarcoma, and metastatic branchitic carcinoma. The new methodology architecture resembles the CNN. Next, survey finding will explain about Lekana.
Survey findings. We find the most important things in this survey. The CNN is the currently most preferred deep learning method for the classification of brain tumor because CNN will generate hierarchical manner of networks. So this is simplest and easy way for classification of brain tumor. Then we can use GPU network architecture in CNN, which can also training the network divides the data easily to classify. For brain tumor classification using CNN via MRI images processing and CNN classification methods aim to identify the tumors and the brain tumor and it classify the tumors as primary and secondary. Thank you.